Wisconsin-Madison. But we have these few apologies from academics today because there is an event, engineering science department event, so we have apologies from Marty and Vinod. So, but despite that, thank you for coming to this seminar today. So today, the Professor Diffee is going to talk about his research. He's a Virginia H. Marsh professor and department chair in the Department of Kinesiology at the University of Wisconsin-Madison, where he also serves as a director of the Marsh Center for Research in Exercise and Movement. And he received his PhD in physiology and biophysics in 1992 from the University of California, Irvine, and has been studying the adaptation of striated muscle cells for over 30 years. So he will be talking about his latest research on contractile function in single cardiac cells.
Thank you. It's a great pleasure to be here. I appreciate very much the invitation. It always sounds weird when I talk about the fact that I've been studying adaptation in muscle cells for 30 years because it feels like I should know more than I know right now if I've been doing this for 30 years. So, but hopefully we'll be able to talk a little bit about some of the stuff that we've learned over the last just more recent years studying adaptation using a particular technique of single-skinned cardiac myocytes. So, all of this work has been done at the University of Wisconsin-Madison. So here's a picture of our campus, a very beautiful campus stretched out across along the lakeshore. And on gorgeous summer days, there's lots of sailing to be had here out on the lake. But of course, Wisconsin doesn't look like this all the time. So this is actually literally a picture that a friend sent me while I was in the air flying here over the weekend. When I landed, I had this picture on my phone of what March in Wisconsin looks like. So you can see why I was glad to get the invitation to come here to Auckland in March to escape scenes like this. And so I do take advantage of the opportunity to escape winter in Wisconsin as often as possible, and it's a great pleasure to be here in Auckland. So I'm not just here to get away from the winter, though. So I want to talk about our experiments using single cardiac myocyte measurements, single-skinned cardiac myocyte measurements, to look at adaptation to three different things that I'm going to talk about. So as Vicki said, I've been interested in studying adaptation to muscle since I was a beginning master's student many, many years ago. But recently have focused a lot on doing single muscle cell measurements because I do feel like, and I hope maybe can convince you that there's some really unique questions that we can answer with single myocyte function that's a little bit difficult to get at in other ways. So the things that I'm going to talk about, so I spent a lot of time thinking about how the heart responds to endurance exercise training. And so I'll talk about some of our work exploring this using single cells. So pictures of exercise always make it look really fun like this, but of course another legitimate form of exercise is what we do in Wisconsin for our endurance exercise training. So, so then we'll switch gears from exercise and talk about some work that we did looking at adaptation to aging. So there's some very specific changes that happen in the heart as we age. And we think looking at changes in cellular function in aging is a really important thing to help us understand this process of altered cardiac function with age. And then really the thing that has brought me here to New Zealand, some work that I've been doing with Chris Baldy down at University of Otago, is really what gets me here to New Zealand, and that's to look at adaptation to diabetes in human cardiac tissue. And this is my favorite description of diabetes is on the math test. Bob has 36 candy bars, he eats 29. What does he have now? Bob clearly has diabetes now. So All right, so let's start with exercise. What do we know about exercise? Well, obviously we've known for quite some time that exercise training is beneficial to cardiac function. Actually known since at least the 18th century that exercise is beneficial to cardiac function. So this is William Herberton, who anecdotally noted in his studies of angina, with respect to the treatment of this complaint, I have little or nothing to advance, although I knew of one who set himself a task of sawing wood for half an hour every day and was nearly cured of subsequent bouts of angina. So we could substitute snow shoveling in here for sawing wood and show the benefits of regular chronic exercise on beneficial effects on this on cardiac function. But fortunately, we have a little bit more specific physiological information. And again, we've known for quite some time that endurance exercise training increases maximal cardiac output. And it does that primarily by increasing stroke volume. So this is work done from more than 30 years ago in rats. Actually, I think this is dogs, where maximal cardiac output during maximal exercise is clearly increased in the trained group, and stroke volume at every submaximal exercise intensity, including rest, is increased in the exercise trained group. So there's been a lot of interest in trying to understand what is this adaptation to exercise that improves cardiac function. We're actually quite used to thinking about adaptation to negative stimuli, so declines in function in response to pathological conditions. So exercise is an interesting intervention to think about because it has primarily positive adaptations in most tissues that people have studied. And that makes it sort of an interesting thing to think about. How does this thing that we do to the whole body actually positively impact things at the molecular and cellular level? So thinking more specifically about the myocardium per se, we know that exercise training increases myocardial contractility, by whatever mechanisms or whatever measures we might use to think about contractility. So positive dPdt, negative dPdt are often indices that we use to talk about contractility. So the rate of pressure development in the left ventricle, the rate of pressure relaxation. Dang it, my thumbs are too big. Here's exercise trained group compared to a control group, and positive dPdt is higher across the board, and negative dP, we have a faster rate of relaxation. ESPVR, in-systolic pressure-volume relationship, this part of the pressure-volume curve here, where if you line up a bunch of pressure-volume curves at different volumes, and draw this slope connecting these dots here, this relationship between pressure and volume in systole. The slope of this line is often given as a nice measure of contractility. And you can see in exercise-trained hearts there's this increase in this slope. So here's the beginning. Here's after 12 weeks of exercise training. There's an increase in this slope, which again is often given as a measure of an increase in contractility or an increase in contractile force in the myocardium, is perhaps another way to think of that. So from the whole heart level or from the whole ventricle level, we know that exercise training improves ventricular function. But the mechanisms for this at the cellular level or at the myocardial per se level have really been difficult to get a handle on. So this is work that spans from the 80s all the way through the early 2000s, where people really looked at pretty much everything they could think of inside the cardiac muscle cell that might influence force production. And most of the things are sort of default is to think about calcium handling by the cell. So people looked at a variety of places inside the cell that were related to calcium handling that might give us an improvement in cardiac function with exercise. So that includes the L-type calcium channel or calcium entry into the cell. That includes the ryanodine receptor or the SR calcium release channel. That includes the sodium-calcium exchanger. And the history of these experiments over the course of 20 years or so was really unsatisfactory in terms of providing any sort of mechanisms for endurance exercise training improvement in function. There really just wasn't, it was really unclear. There was some positive experiments that seemed to indicate that there was a change with exercise training that would support an improvement in contractility. Many studies that saw no change with exercise training. Of course, the bottom line of all of these experiments was the suggestion that as a result of exercise training through whatever mechanism, you ended up with elevated intracellular calcium during activation in the heart, and that's what gave rise to increase in force. So when it became much more commonplace to be able to measure intracellular calcium, several groups took it upon themselves to measure intracellular calcium in single myocytes. At the same time, they were measuring shortening, and do this in cells from exercise-trained hearts compared to sedentary control hearts. And lo and behold, found absolutely no evidence to indicate elevation in intracellular calcium as a result. So here's our sedentary hearts, here's our trained hearts. Here's no change in calcium, even though there seems to be some changes in shortening properties. So a somewhat nice resolution to this idea of why couldn't we find anything at any of these subcellular sites having to do with calcium handling that indicated an exercise effect. It's because there's ultimately no real evidence to indicate that calcium handling is affected by exercise training. So if you eliminate all of these sites that might ultimately impact calcium levels inside the cell during activation, a sort of likely thing that's left over is there a change in the way the myofilaments respond to calcium? And that's where we stepped in to begin to address this idea of, is there some change in myofilament response to calcium as a result of exercise training? And I feel like this is an aspect of myocardial cellular physiology that single-skinned cardiac cells are sort of uniquely positioned to help us answer. So really the rest of the experiments that I'm going to talk about here today are really based on these single cardiac myocyte, skinned, membrane removed, that means, and attached to an apparatus that can measure force, shortening velocity, power output. And what this allows us to do is put the focus on the contractile apparatus. When we skin our cells, everything else goes away in the cell. Mitochondria go away, SR go away, nuclei go away. And what we're really left with is just the myofilaments, just the contractile apparatus. And so if we're interested in looking at myofilament adaptation, it sort of helps to get rid of everything else except the myofilament, and then we can address whether there's actually a change at the myofilament level. Skinning the cell also gives us control of the intracellular environment, so we can control calcium and look at the sensitivity to a given level of calcium activation. But unlike test tube prep, we actually keep the myofibrillar structure intact, which seems to be important. I think there's a history in muscle physiology of sort of losing focus on the exquisite array of protein arrangements relative to each other that is true in an intact cell versus grinding up proteins and looking at them in the test tube. So we really want to keep the arrangement of myofibrillar proteins intact, and that's what our skin cells allow us to do. And then we eliminate the complexities of some of the multicellular preps that I know lots of people here in the room are used to using. So we really like the idea of we have easy diffusion in and out of the cell. We know that the protein component of this cell is way simpler than the protein component of a large multicellular prep. So we just, prior to us doing this work that I'm about to tell you about, skinned cardiac myocytes had been used routinely to just look at basic cardiac physiology, to really trying to understand the role of various contractile proteins, various regulatory proteins. but it hadn't really been used as a technique to study adaptation to something like exercise. So we took it upon ourselves to do that. So our exercise training program, which we've developed over the course of years, so this is done in rats. These studies I'm about to tell you about are done in female Sprague Dawley rats. I don't know to what extent you care about the rat exercise training program, but what we did is an hour a day, 5 days a week, forced treadmill running. So here's rats running on a treadmill. Female Sprague Dawley rats at young age really like to run. Male Sprague Dawley rats really don't like to run, which is why we use female Sprague Dawley rats. Old rats of any sex really don't like to run, so I'm going to tell you about some aging studies, and I'm definitely not going to show you pictures of old rats trying to run on the treadmill because it's not a pretty sight. But these guys will really run pretty willingly, so it's actually a pleasure to do exercise training of these rats. So this is a relatively high intensity exercise protocol. 26 meters per minute is pretty fast for these guys, and a 20% grade just to make it really hard for them. And we do this over the course of 11 weeks. And it was important to us to look at evidence of training effect. We wanted to make sure that these guys really were adapting to exercise. So a modest increase in heart mass is a pretty good hallmark of an endurance exercise training effect, and we saw that. Increased heart weight to body weight ratio, we saw that, and an increase in one of the mitochondrial enzymes in Krebs cycle, citrate synthase. In skeletal muscle, that's also a pretty common hallmark for our endurance exercise training effect. And then we wanted to do this other step, which is to actually test their exercise capacity. Now, this is even a more robust test. This is now where we're really pushing them to run at their maximum exercise speed. And we basically let them run until the exhaustion, until they can't run anymore. And the distance that they run before they're exhausted is a measure of their exercise capacity. And we saw a very strong increase in exercise capacity in these guys. All right, so we did of this 11 weeks of exercise training. Then we had a sedentary group that got off easy and didn't have to do any of the exercise. And then at the end of that, we took the hearts out and prepared skin cardiac myocytes. So I just want to lead you through our procedure. One of the really important things that we established that was actually crucial for us to do pretty much everything I'm going to tell you about today is that we can do this in either fresh hearts just removed from the animals, or we can do this in frozen tissue. So we can take the hearts out of the animals, freeze them in liquid nitrogen, store them at -80, and then come back at our leisure to do our cellular measurements. So this is crucial for studies of adaptation, because if we're going to do 10 animals in the exercise group and 10 animals in the sedentary group, we kind of need to do all the exercise at once, take out the hearts, freeze them, and then start doing our cellular measurements. So the fact that we can do this in frozen tissue is crucial, and actually turns out to be very crucial in the stuff that I'm going to talk about at the end, the stuff that I've done with Chris in the diabetic tissue. So take the hearts out, trim them, trim off the atria, trim off the right ventricle, so we're just left with the left ventricle, and then we either freeze it, or on the fresh tissue, if we do it in on the frozen tissue, we take it out of the freezer, let it thaw, and then mechanically disrupt this ventricular tissue. So mechanical disruption is really just a fancy way to say we mince it with scissors and then we throw it in a blender. And literally we're just trying to disrupt the tissue in the blender, and what we end up with is coming out of the blender for really just a few seconds in the blender is enough to give us a mixture of junk, chewed up cells, big globs of ventricle that are still left over, but somewhere in there are single myocytes that have been disrupted away from the rest of the ventricular mass. What we've come to call this technically is single myocyte sized preparations, because we really can't guarantee that this is actually just one myocyte. It could be two myocytes stuck together that look like one myocyte. We don't do anything to disrupt those. As long as it's roughly the size of a myocyte, we're pretty happy about that. So then we wash these, and then we skin them with a detergent. Triton is our detergent that we use, and this is what dissolves all of the membranes associated with this cell. And ideally, we're left with some really pretty looking cells that look like this. And then we select a cell that looks really nice to us, and then we attach that to our force measurement apparatus, which consists, like many of you are using in here, a force transducer that can measure, has a sensitivity enough to be able to measure force of a single cell. So we use Aurora transducers that are these capacitance transducers, sensitive enough to measure the force of a single cell, and then a motor positioner that allows us to control the length of the cell. I won't go into our attachment technique here. I'm happy to talk about that afterwards for anybody that's interested. We use a technique that we use for skeletal muscle cells, because when I first started doing this, I was broke and couldn't really afford a skeletal muscle setup and a cardiac muscle setup, so I just wanted to do both of these on the same setup, so I just adapted our skeletal muscle attachment, skeletal muscle fiber attachment technique to be useful for cardiac muscle. All right, so once we have our cell attached, then we can just go through a protocol to measure force. And our idea was that there would be a difference in the sensitivity of force in these myocytes, a difference in their sensitivity to activation by calcium. So here's an experiment that we do to look at calcium sensitivity. So when you're doing these single myocytes, we cannot rely on just absolute force measurement when we move it into a different bath, because the surface tension of the solution is roughly the same order of magnitude as the force that we get out of a single cell. So just moving it from one chamber to another gives us a huge force change that may have nothing to do with the active force in the cell. It may just be the difference in surface tension from one chamber to another. So what we do to more precisely measure force is put it in a chamber of a given calcium concentration, let it get to steady state force, and then we slack that cell using our motor positioner, doing a slack test, which is a standard muscle physiology technique that's been around for a long, long time. When we slack that, it goes to 0. And then you can see it starts regenerating force. What we're interested in, though, is the difference between the force at slack down here and the difference in force right before the slack. And that's going to be our active force in that solution. So we put our cells in a given PCA solution, slack it, move it back into relax, measure that force on our scope, then put it into a different PCA solution, et cetera. So here's our just a representative experiment where we do, so PCA is just like pH, it's the negative log of the calcium concentration. So a PCA of 4.5 is 10 to the minus 4.5 molar calcium. So PCA 4.5 is generally considered maximally activating calcium. PCA of 9.0 is generally considered the calcium concentration in a resting unactivated cell. So we do a variety of different PCAs, measure the active force. Here's a single cell from control hearts. Here's a single cell from trained hearts. And what we end up with is this tension PCA relationship, or force PCA relationship. And so we just plot the force at each PCA. It fits nicely to a sigmoidal relationship, which is pretty classic for striated muscle. And then so several different ways of looking at this. One is to just look at the force at any given calcium concentration. And you can see that trained PCA 6.0 here, a trained cell has higher force than the control cell. Or to look at the position of this curve. So we say increase in calcium sensitivity means a left shift of this tension PCA relationship, and that's what we're seeing here. Did a lot of work to generate these curves. So this is over 100 cells, actually per group, 100 trained cells and 100 control cells. And I got tired of people looking at this and thinking that this doesn't mean very much in terms of adaptation, because there's a small little difference in the position of this tension PCA curve. So I like this way of showing at submaximal calcium concentrations the really significant improvement in calcium sensitivity that exists here. So at PCA 5.9, we have almost 60% of our maximum force in the trained cells, whereas we were less than 30% of the maximal force in our control cells. So for a cardiac cell that spends a lot of its life at submaximal calcium concentrations, this difference in calcium sensitivity is huge in terms of the amount of force that the cell can generate during the course of a cardiac cycle. So just to summarize what I just said, exercise-induced increase in calcium sensitivity provides a mechanism for increased force without an increase in intracellular calcium. Since people had not seen an increase in intracellular calcium, but had seen an increase in force, this change in calcium sensitivity provides a mechanism for how that might be possible. But as cool as I think doing tension PCA relationships are on skin cells, it's important to recognize that these are isometric contractions. So we attach the cell between the force and the motor, and we're letting the force generate, get up to steady state in an isometric condition. We're not letting the cell shorten at all. I just think it's important to realize that this isometric contraction with no shortening, there's only a small part of the cardiac cycle that is represented by isometric shortening. So here's a pressure volume loop. So here's filling of the ventricle. Here's the start of contraction. We have this isovolumetric contraction phase, and then we have this ejection phase, and then relaxation, and then refilling, et cetera. And it's only this part of the cardiac cycle that really is represented as isometric contraction of the myocardial tissue. So that's interesting and all, but really if we're talking about the ability of exercise training to improve cardiac function in a way that improves ejection of blood from the heart, this isometric contraction is really not telling us how the heart does at actually pushing blood out of the heart. So there are some preps that do allow shortening. Again, as many of you know in here, looking at free-floating myocytes, electrically stimulated intact myocytes, we get shortening, but it's important to recognize that this is unloaded shortening, so there is the cell doesn't have to overcome any load in order to shorten. So what we wanted to do in order to approximate this actual shortening, which is shortening of the myocardium, but shortening against a load in order to eject the blood from the heart, was to look at loaded shortening properties. And so this is how we did this. So this is, again, our cell attached between our motor and our transducer. Again, put it in whatever calcium concentration we want it to, allow it to generate steady state force. And then at this point, we use our motor and our software to step to a specified force, something less than maximal force. And then when we step to that force, in order to hold that force constant, the motor allows the cell to shorten in order to keep that force constant. And then step down, back down to slack force, do another activation, and now step to a different submaximal force, and each of these submaximal forces is associated with shortening at a particular velocity. And so then if we take the slope of this length change versus the force, we can construct a force velocity curve. So here's our different forces, our different submaximal forces, and here's the velocity of shortening at each one of those submaximal forces. So this force velocity curve has this pretty characteristic hyperbolic relationship, which has been seen at pretty much every level that people have studied shortening and force in a muscle prep, all the way from single myofilament all the way up to a whole muscle. We have this pretty characteristic relationship between force and shortening velocity. So we did force velocity curves for cells from sedentary control hearts and from exercise trained hearts, and they look like this. So here, open circles are our trained guys, closed are our control. So what we saw is a increased velocity at any force across the force velocity curve. So our trained cells shortened at a faster velocity at the same force compared to our control cells. So again, this looks a little bit disappointing given the number of cells that we did. It just doesn't look that impressive. But what we wanted to do was to really think about what does this mean for the cells in intact ventricle as they're pushing it against the blood, trying to eject the blood, and recognize that if you take each one of these points, this is a unique force times a unique velocity. The product of force times velocity is power output. And now we're getting somewhere. Now we're getting to think about the work output of the myocardium during a cardiac contraction. How much power, how much work per unit time can the myocardium do during a cardiac contraction? And now you can see that our peak power output in our trained guys is really elevated over our peak power output in our control guys. So we're seeing some sort of molecular cellular adaptation that allows the cardiac myocyte to generate more power during a contraction as a result of exercise training. All right, so then we were interested in looking at what are the molecular mechanisms for this exercise adaptation. So I'm going to skip through many, many years of failed experiments here because we really looked at a variety of different molecular mechanisms and ended up with some very unsatisfying answers. One of the advantages, again, of doing skin cardiac cells is we've gotten rid of a whole bunch of stuff in the cell. So if you see a difference in behavior between cells of different types, you can be pretty sure that that difference is somehow connected to the myofilaments, because that's all that's left in these cells. So it does sort of streamline our looking for a molecular mechanism, because we know the answer is in here somewhere. But of course, there's still lots to look at here. So a variety of different things that could be responsible for our increased calcium sensitivity and increased power output. Protein isoform changes are a likely explanation. So these troponin subunits, troponin T, troponin C, troponin I, all come as different isoforms. We thought it was possible that there was an isoform shift as a result of exercise training. Turns out that's not the case. We couldn't see any evidence for isoform shift. Subunits of myosin, the myosin heavy chain and the myosin light chains, all come as protein isoforms. So we looked at myosin heavy chain. We looked at some myosin light chain changes. Spoiler alert, we missed a crucial one because we didn't know how to look at it. But we didn't see any evidence of any effect there. In addition to protein isoform chains, a number of these myofilament proteins are phosphorylatable, namely cardiac TNI, myosin light chain 2, and myosin binding protein C. These are all phosphorylatable as a result of adrenergic stimulation or other hormonal stimulation. So we thought maybe there was altered protein phosphorylation levels. It turns out there wasn't any evidence for altered protein phosphorylation as a result of exercise training. So we were stumped a bit about what is the molecular mechanism. Again, we felt like the answer was here somewhere, but we didn't know how to look for it. So this was several years ago when microarray or gene chip technology was first becoming available. So we jumped on that bandwagon, and I'll skip over a bunch of our experimental methods. But ultimately, we looked at the expression of a whole bunch of genes simultaneously, and we compared the expression in trained hearts compared to control hearts. So here's all the genes that were different, decreased expression, increased expression. two things that were interesting to come out of this. Everything in red here is genes that had previously been found to be upregulated in another model of cardiac adaptation, that is pathological hypertrophy. So we thought that was interesting. But then the other thing that struck us, actually the only one on this whole list that's associated with the myofilaments, was atrial myosin light chain 1. It had not dawned on us to look at atrial myosin light chain 1 because this is a ventricular preparation. And turns out it's really hard to look at. We did go back and now do the protein work, which you have to do by 2D gel electrophoresis, where you separate out the ventricular isoform and the atrial isoform in this two-dimensional gel, where you first separate by isoelectric point and then separate by size. So here's a preparation from the atria. Here's our atrial myosin light chain. Here's a preparation from the ventricle. Here's our ventricular myosin light chain. If we add atrial tissue and ventricular tissue together, we can see both the atria and the ventricle myosin light chain together. And here's our trained myocardial tissue, and we see the presence of atrial myosin light chain, unlike in our control where we see no evidence. So that was kind of cool. We found both at the gene expression level and the protein level that training did seem to result in an increase in atrial myosin light chain. So we went back to our literature to figure out what the heck is going on with atrial myosin light chain. So first of all, so the light chains are these small little subunits on the myosin molecule that people have been trying to figure out for years. What do they actually do to modulate contractile function? It turns out in the heart, there's two different isoforms of this essential light chain, the atrial and the ventricular light chains. And the atrial form is the embryonic form. Across the heart, everything expresses atrial at the embryonic stage. But then as the heart matures, the ventricle switches to ventricular, the ventricular isoform. Atria continues to express the atrial isoform. But we found some evidence to indicate that in these pathological conditions like hypertrophic cardiomyopathy or hypertension in a pig model, that atrial isoform is re-expressed. And this re-expression of atrial isoform is associated with an increase in calcium sensitivity and an increase in power output when the atrial isoform is present. So we feel like we tied this up in a kind of nice little bow. So we found at the cellular level, so I didn't talk about this, but no effect on maximal tension. But we did see an increase in calcium sensitivity of tension, that is greater tension at the same submaximal calcium. No increase in maximum unloaded shortening velocity, but an increase in loaded shortening velocity or increase in peak power output. And we felt really good that our increase in atrial myosin light chain expression and protein expression could provide a molecular mechanism for this. All right, I want to switch gears, talk a little bit about aging. So as we tried to follow up on this work and went to get this grant renewed, the kind folks at NIH said, gee, it really seems like you've kind of solved this, and there's really not much more to learn about exercise except some really incremental improvements in our understanding. So we started thinking about, well, what's a thing that happens to the heart that's a bigger effect that we could begin to address, still thinking about exercise. So we thought about the aging heart, which does have this, a number of cellular changes, probably the biggest of which is a loss of cardiac myocytes. So we lose cells as we age. Those cells are replaced with connective tissue. So our ventricle gets stiffer, and that has some huge implications for cardiac function. But in the cells that are left, we do have evidence of a decrease in contractility or decrease in contractile function along with the loss of cardiac myocytes. So we wanted to study that at the single cell level and actually look at the effect of exercise on that. So we focused on force velocity and power output relationships on these, and we looked at 3 different ages of rats. So this is a hybrid strain of rats that was specifically developed at the National Institutes of Aging to study age-related changes in cardiac function. So they lived to about 36 to 39 months of age, so a 33-month-old rat would be roughly the equivalent of a 75-year-old human. 24 months is older adult, maybe the equivalent of a 40-year-old human, and 9 months is a young adult. So we isolated myocytes from hearts of 9-month-old, 24-month-old, and 33-month-old rats, and you can see there wasn't much difference. We'll just focus immediately on power output. There wasn't much difference between the peak power output between 9 months and 24 months, but there was a big drop in peak power output associated with aging to 33 months. And we put these guys on the treadmill to see if we could mitigate any of these effects with exercise training. So we did 2 different intensities of exercise training, mostly because these old guys couldn't really do exercise the way the young guys could. So we had to modify our exercise protocol. So we had a sedentary group, and then a moderate intensity exercise group, and a high intensity exercise group. Interestingly enough, the moderate intensity was the only exercise that actually improved peak power output in these myocytes from these guys. High intensity exercise didn't look any better than sedentary. So we're following this up to look at some molecular mechanisms. We don't really have any candidates at the moment. So just summarize our loaded shortening velocity changes and our maximum power output decrease in 36-month-old guys. I don't know our molecular mechanisms, and exercise training minimizes some of these age-related effects. So I want to take just a brief detour here and talk a little bit about, so I've tried to keep a foot in both cardiac muscle physiology and skeletal muscle physiology because I'm interested in adaptation to both of these muscle types. And I just want to talk about some of our stuff in age-related skeletal muscle fiber function because it sort of relates to what we're going to end up doing in this diabetic project. So we've done single skeletal muscle fiber, tension PCA, and shortening force velocity curves and power outputs, and we see something similar, that there's a decrease in calcium sensitivity as we age in skeletal muscle, and a decrease in peak power output as we age in skeletal muscle. But the reason I want to talk about this is that we partnered, in order to look at molecular mechanisms of this, we partnered with a group at University of Wisconsin-Madison that is really pioneering some great techniques in proteomic analysis, where we can just do our contractile measurements, give them the tissue, and they can analyze proteins across a huge number of proteins to look at changes in both protein expression and post-translational modifications like phosphorylation or other changes. When we did this, so this is the same sort of tissue, our 6-month, 24-month, and 36-month tissue, we saw a change in phosphorylation state of the regulatory light chain, where there was a decrease in the amount of phosphorylated regulatory light chain, and a shift towards the unphosphorylated form of our regulatory light chain as we went across the age spectrum. So this has led to a really nice partnership that, again, we're going to take advantage of with the diabetic hearts, where we can really explore these functional changes by combining our mechanical measurements in our single cells, whether it's single skeletal muscle fibers or single cardiac myocytes, with this pretty sophisticated proteomics information, and begin to correlate changes in function with changes in proteomics that end up resulting in the phenotype that we're seeing at the whole animal or whole human level. So we'll come back and talk about this in just a moment. So here we are, just a moment. We're going to, I want to talk about this project that is really what led me to start coming to New Zealand on a regular basis to get away from the snow. So we're exploring this declining cardiac function associated with diabetes. And again, I think I have to belabor this point to too many people in this room that diabetes is associated with declining cardiac function with a variety of indices that we might use to describe cardiac function. This is work that I didn't do at all. This is work that the group down at University of Otago with Jeff Erickson and Chris Baldy has done, looking at now at the trabeculae level our diabetic and non-diabetic control, looking at this decline in force intrabecularly at different stimulation frequencies. Again, a decrease in force generating capability at least the trabecularly level. All right, I'm going to skip through some of this, but again, default thinking is if there's a decline in force, maybe calcium handling, altered calcium handling is the culprit. In the group at Otago's hands, there didn't seem to be a change in calcium handling, at least as measured by the amplitude of the calcium transient or by the SR calcium load. Didn't seem to be a difference between the diabetic and the non-diabetic. So again, it's sort of the same process that we ran through with the exercise training, except the opposite problem. Now we have declining cardiac function, but not really any evidence indicating dysfunctional calcium handling by the calcium handling apparatus within the cell. And so, just like with our exercise training, it led us to think about, well, is there a myofilament explanation for this declining force independent of what's going on with calcium? So, if we are interested in myofilaments, skinned cardiac myocytes come to the rescue because that's a really nice way to look at myofilament changes. So here's our project that we started a little while ago. We're actually right in the middle of doing this right now. So, but this is now ventricular biopsies taken from human subjects that are undergoing some cardiac procedure at the University of Otago. So from my standpoint, personally, this is pretty exciting for me because I've spent my entire career looking at animal models, usually rats and mice and rabbits, looking at skeletal muscle or cardiac muscle preps from these animal models. So this is a really nice step forward to think about doing some of these same measurements in tissue from human samples. I know those of you who do human subjects research is not a big deal, but for me this is a pretty significant step forward to think about moving from a rat to a human and still getting some cellular information about this. So the other nice thing about taking this from human subjects that are undergoing some other cardiac procedures, we have a lot of data about their cardiac function. So just there's a lot going on here, most of which we don't need to pay attention about. I just want to point out one important parameter is both of these groups. So here's our diabetic groups, here's our non-diabetic group. Both of these have preserved ejection fraction. So we can have diabetes progress to heart failure to the point that ejection fraction is compromised. And we certainly have non-diabetic progression to heart failure through other mechanisms. That's a completely different story that we might want to look at some point in the future, but right now we're looking at diabetic with preserved ejection fraction compared to non-diabetic, also with preserved ejection fraction. So we're really just looking at specifically the effects of diabetes. All right, so when these biopsies are taken while the subjects are on the table, they're immediately frozen in liquid nitrogen and then stored. So here we're very much taking advantage of the really happy information that we can do this analysis on frozen tissue. So we can store this tissue. I think Chris, correct me if I'm wrong, I think you guys have a tissue bank of tissue from over 300 subjects on hand there at University of Otago. So for the beginnings of this project, Chris sent me some frozen samples, shipped them up to me at University of Wisconsin, and I proceeded to isolate single cells, skin them, and measure the calcium sensitivity in these diabetic and non-diabetic human cardiac samples. So here's our results. We have our diabetic with preserved ejection fraction, non-diabetic with preserved ejection fraction. So now we're seeing a right shift in the tension-PCA relationship, so indicating decreased calcium sensitivity of force. So again, another way to look at this is just choose a calcium level and see that we have less force at that calcium in the diabetic tissue or the diabetic cells compared to that same calcium at the, in the non-diabetic cells. We're looking for a way to characterize the calcium sensitivity of these cells. One common way to do this is to identify what's called the PCA50. So that's the calcium concentration at which we achieve 50% of maximal tension. So we just kept track of the PCA50 for all the cells that we did. And there's a couple of different ways that we can express this. So here, each symbol here is an individual cell that we did. And then just kept track of whether it was a cell from a non-diabetic subject or a cell from a diabetic subject. One of the really interesting questions to think about and debate in single cell measurements is the cell the functional unit, or is the cell just representative of what's going on in the whole animal or the whole person? So if you think about experimental design, you could think about our N is actually 4 subjects per group for non-diabetics. So here's the mean of all the cells that we did from this guy, from this guy, from this guy, from this guy. So maybe that's the best way to look at the data, is to group all the cells from one individual together and actually express that as the mean from that individual. So there's schools of thought in both directions, because diabetes is happening to the whole person, not to the individual cell. On the other hand, our measurement is at the individual cell level. Fortunately for us, the answer came out the same regardless of which analysis we did. That is, there's a decline in PCA50 in the diabetic tissue. Sorry. So indicating a mechanism for the decreased force generating capacity of the myocardium because we have decreased sensitivity to activation by calcium. And this gives us a way to think about force independent of what changes might be happening in calcium handling by the cell, because there's decreased calcium sensitivity. All right, so here's what I just said. Decreased calcium sensitivity in cells from subjects with diabetes compared to non-diabetic. And these are diabetics with preserved ejection fraction. So we're currently working on mechanisms right now, and here's what I think is going to be very exciting. So we're again partnering with my collaborator in the University of Wisconsin to do proteomic analysis of this very same tissue that I've gotten from Otago. So I split this tissue into half. I isolated cells from half of the tissue, sent the other half to do proteomic analysis on this. So we're very interested now to see protein changes in the very tissue that we have cells from, to really, hopefully really begin to explore what is the molecular mechanism for this change in calcium sensitivity. So hoping for some exciting results on that. All right, I'm out of time here, and so I just want to thank the people who actually did the work. These are grad students in my lab over the years that have done most of the work of sitting down in front of the microscope attaching cells. And so I appreciate all their help. And then I had some great collaborators at UW-Madison group that helped us do the microarray expression and the protein work, Jeff Johnson and Tor Stein. And then here's our proteomics, Inga, her grad student Zach and her grad student Yutong have been helping us with doing the proteomics analysis. And then of course the folks down at University of Otago, Chris, who got this whole project started, and then Jeff and Regis, who have been doing a lot of the trabecular work and are also contributing to our thinking about this. So I think I left a few minutes available for questions, and happy to have any questions that you guys might have. Yes?
Hi, Gary. Nice talk.
Thank you.
So I have a question about the shift that you saw in contraction velocity in your myocytes.
Yeah.
So I'm a skeletal muscle person.
Okay.
So what that looks like, so much from skeletal muscle is a shift from type 1 to type 2 distribution. So just because I don't know much about cardiac muscle, is there an analog in cardiac muscle, and does that point more so to your hypothesis about myosin heavy chain?
Yes, a great question, and I didn't show our data. So yes, there's two myosin isoforms in cardiac muscle, alpha versus beta. Beta is the equivalent of slow myosin in skeletal muscle. So I don't know if you're talking about the exercise shift or the aging shift or both. So exercise was not associated with a shift in myosin heavy chain isoform. So we kind of thought it might, looked at that right away, but there was no change. Aging, the slowing of velocity, is associated with a shift towards more beta. And that's problematic in the translation of this animal work to human, because there's no indication that humans really change myosin isoforms under pretty much any conditions. So any effect that we see that's due to a shift in myosin heavy chain is going to be unique to animals and not really applicable to humans. So maybe I'll just go there real quick, if I can do anything real quick here. The one thing that the one thing that we think is really not it's evidence that myosin heavy chain shift is, at least if it's important, it's not the only thing that's happening, is you get a pretty substantial shift from 9 months to 24 months in myosin heavy chain, but not really much of a change in peak power output or maximum shortening velocity. And then a much more minor shift in myosin heavy chain as you go from 24 to 36 months, and that's where we get the biggest change in shortening velocity parameters. So we certainly can't discount the role of myosin heavy chain, and it is the driver for shortening velocity. So obviously a shift in myosin heavy chain isoform is going to drive this, but there seems to be at least something else going on if myosin heavy chain is an important part of this. But certainly our exercise effect cannot be explained by myosin heavy chain because we saw no shift whatsoever with exercise. So, yeah. Yeah, Andrew.
Thanks, Gary. Cool stuff. I was really encouraged to see that moderate exercise was better than intense. Why do you think that's?
Yeah, great question. So I think the field of aging and muscle function, whether it's cardiac muscle function and skeletal muscle function, is littered with groups that have done 1 or 2 aging studies and then never ever done any aging studies again, because it's really difficult to interpret this. And I think I might be in that group, because when it comes to exercise with both, so I didn't show our skeletal muscle work, exercise is horrible for aging skeletal muscle as well. It makes the muscles worse. So our theory, that I don't know if we'll ever get around to testing, is that there's a certain sort of level of functional and structural quality in the muscle that can tolerate exercise. And if you're sort of below that level, exercise is really beneficial. If you get above that level and the muscle is starting to fall apart, exercise just pushes the cell over the edge. And so our high intensity exercise, not only did cellular function not really improve, our loss of myocytes was actually escalated by the high intensity exercise. Again, the idea being that if cells are sort of teetering on the edge of life or death, high intensity exercise just pushes them over the edge. And the same thing we found was with our skeletal muscle. We really hope this whole project was really designed to study effective exercise to rescue skeletal muscle mass, and it didn't do it at all. In fact, it actually made it worse. So I think there are some lessons in there for overall exercise physiology, although I always caution people against trying to draw specific correlations to rats running on a treadmill compared to humans running, because they're very, very different physiological events, I think. But I think it is interesting to think about exercise as not either just an on-off switch. There is some calibration of the exercise effect that I think it's important for us to think about. Yes?
I've got a methods question.
Yes?
I find it quite interesting that you guys freeze your tissue
Yep.
and thaw it. I've never actually heard of someone getting cells from that. How does that work? Do you guys engineer to just it?
Nope. So this is all still, the technique is the same whether we start with fresh tissue versus frozen tissue. And this is actually the very first thing we did before we started our exercise training, because I didn't want to have to spread out the exercise training among all of our different rats so that we could have a fresh rat every day. So I thought, well, if we're going to do this, we're going to have to try this in frozen tissue. So I think that what we're saved by is skin cells. We've gotten rid of all the things that are really, really prone to damage from freezing. So membranes are gone, ion channels embedded in the membrane are gone, mitochondria are gone. So what we're really left with is just the myofilament proteins that are sort of held still in this arrangement. But that's not nearly as prone to freezing damage as all the other enzymes and other more sensitive proteins is. So that's my theory about why we can get away with freezing. Yes?
Hi, Gary. Sorry, maybe I missed something, but in the human study, are these biopsies, from atrial tissue?
No, sorry, this is ventricular biopsy tissue. So I think they also do atrial appendage stuff, but this is actually ventricular biopsy tissue where they go in and take a little piece of the little wedge of the ventricle. So it's very small. I mean, 15 milligrams is the amount of tissue that we get. I don't need to denigrate the contribution of the people that gave up 15 milligrams of their heart tissue so that we could do this, but I always wish there was more. But yeah, it's a very tiny amount, a tiny piece of the ventricle that's taken out of the subject. So.
Do you think like we'll have some differences from the atrial myocyte?
Oh, I'm almost sure that there is. I think everybody that's ever looked at atria versus ventricle, they're very, very different tissue at the protein level. So I suspect there would be. I think most of us that are interested in adaptation are more interested in the ventricular adaptation because that's really what drives systemic circulation and the problems or the benefits that come from a positive or negative adaptation.
Okay, so maybe we can continue the discussion over this drinks and nibble because it's over part 5 here already. So let's thank Professor Diffee again and then.
Thank you.